Alright, so this one, okay, revise again. So the idea of a source and a sink, okay, this one is in topic transport in plants, right? Okay, so sugars are transported within phloem sieve tubes from a source such as the mature leaf to a young leaf, which acts as a sink. Now, what you need to know is that what do they mean by a source and a sink? So, a source, by definition, okay, it is any, okay, part of the plant, okay, part of the plant, okay, that actually photosynthesize. Okay, so asalya has the ability to photosynthesize, that immediately becomes a source. Alright, now whereas the sink, okay, is defined by the part of the plant that, okay, uses sucrose or uses the product of photosynthesis for what? Contohnya use it for, okay, for growth. Okay, contohnya in any growing parts of the plants like fruits, seeds, flowers, or it is also can be stored as well. Storage. Okay, kalau storage, we know that sucrose will get converted into starch. Contohnya, in this case, di mana? In the roots. Okay, that's an example as well. Alright, so make sure you know and you understand what do we mean by a source and a sink. So the young leaf also needs water and dissolved mineral ions which arrive at the leaf within the xylem vessel. Alright? Okay. As the young leaf matures, the quantity of the sugar decreases to 0, but the need for water increases. Now, what does that mean? Okay. When the young leaf matures, alright, so awalnya it was a sink. Okay. But as it matures, it now has the ability to photosynthesize. Kenapa it can photosynthesize? Because the structure has already matured. That means it will have all the organelles that it needs, for example, chloroplast, okay, etc., etc. Okay, so once they become a mature leaf, they have the ability to produce their own sugar. So that's why sugar makin lama decreases and in the end 0. Because why? They don't need to uptake, they can make their own sugar. But as they grow mature, water intake increases. Now, how do we link to that knowledge? Because going back to why is water needed? Water is needed for photosynthesis. So if the young leaf already mature, it can start to photosynthesize. Water is the substrate for photosynthesis. So when there is increased rates in photosynthesis, there is increase in the water uptake that is needed for photosynthesis. That's why water increases. Okay, now why is it has the ability to increase? Kenapa photosynthesis rate increases? Is because as the young leaf punya structure starts to mature, it fully developed, it has a lot of stomata as well. Okay, lagi satu. So not only we're talking about photosynthesis, the next one is that it has more developed structure. There's going to be more stomata. If there's going to be more stomata, that means there's more water going to be diffusing out, water molecules or water, sorry, let me correct it again. Water vapor would diffuse out via transpiration through the stomata. So that's why makin lama, the need for the water will also increases. Okay? Okay, now please read through the answers that I have written here, okay, including all the points that I've just described. As the young leaf matures, it gains the ability to photosynthesize. Therefore, it will not need to take up sugar because it can produce its own sugar. That's why, okay, the leaf decreases to 0. At the same time, okay, the need for water increases because plant now needs more water as there is more photosynthesis. Okay, remember, okay, water is the substrate for photosynthesis. Okay, substrate for photosynthesis. Okay, Now, also know that plant leaf structure has matured. There's going to be more stomata available di mana in its epidermal layer. That means that the rate of transpiration, which is the loss of water vapor through the stomata, increases, which leads to even more water uptake that needs to replace the water that has been lost. Okay? For this one, right, this one is relatively simple question, which is asking us to recall the features that is present, okay, in our plant transport tissues, which include companion cell, phloem, and xylem. Now, I hope everybody remembers that companion cell is always going to be found right next to the phloem sieve tube element because it serves a function that helps with the that helps with transport of sucrose, okay, that happens in the phloem. All right, and of course, when we are looking at this under the microscope, you should expect that the phloem and the xylem are going to be grouped relatively close together because they are usually in a bundle. Okay, di mana? Okay, remember our vascular bundle. Okay, vascular bundle. Okay, so if you have this practical and you are asked to identify where can we find, okay, the xylem and the phloem, they should not be too far away from one another because they should be in the vascular bundle, phloem right next to the xylem, and of course companion cell will be always next to the phloem. Alright, let's go through the features di sini. Okay, so cytoplasm. Okay, cytoplasm is going to be present in the companion cell, yes. It's going to be present in the phloem sieve tube element, yes, but it will not be present in the xylem vessel because it's totally empty. Right? However, in the phloem, the cytoplasm is actually very little. Okay, take note. Okay, cell surface membrane. Do they have cell surface membrane? Yes. Okay, the companion cell will have a cell surface membrane. Phloem sieve tube element will have a cell surface membrane, but xylem vessel will not have a cell surface membrane. Kenapa? Because the xylem vessel, they are dead cells, whereas companion cells and phloem sieve tube elements are living, so it still has the living components of its parts of the cell. Okay, cell surface membrane, right? Lignified cell wall. Okay, lignified cell wall. Companion cell will not have lignin, right? Phloem sieve tube will not have lignin, but xylem does have lignin. Make sure you remember the function of lignin, okay, because lignin provide, okay, support, okay. It supports the xylem, okay, withstanding the hydrostatic pressure as water is transported up through the xylem, okay, preventing the xylem from breaking, yeah. And then last but not least, nucleus. Okay, the most obvious one that we need to remember, okay, xylem vessel, yeah, will not have any nucleus. It does not have any contents or whatsoever, no organelles, no cytoplasm, no nucleus. It is hollow. Okay, phloem also will not have nucleus because it will make space. Okay, it needs the space for the transport of sucrose, phloem sap. Okay, however, the companion cell will have nucleus because we know that the companion cell supports the phloem in terms of the function, especially when it comes to, okay, sucrose loading. Okay, sucrose loading that occurs between the companion cell, okay, and the phloem sieve tube element. Okay, and then recalling back that in the xylem vessel it should not have a nucleus because it should be completely hollow. Why is it important that it's hollow? It allows for our continuous undisrupted flow of, okay, water, okay, and mineral ions of the xylem. Of course, okay, another thing that we need to remember, what ensure continuous flow and undisrupted flow ani as part of our syllabus. Okay, again, just a quick reminder for everybody. Okay, I want you to remember the, okay, the cohesion tension theory. remember that one? Okay, so to help with that, okay, remember again, just for the sake of revision, okay, what is cohesion and tension? Okay, cohesion and tension theory of mass flow of water up the xylem. So why water has cohesion? Because water is cohesive. Oops, let me just spell that one. Water is cohesive. Okay, cohesion is whereby, okay, water molecules actually forms hydrogen bond with one another. Hydrogen bonds with one another, with each other. Okay, that's cohesion. Okay, the next one is adhesion. Okay, whereby, okay, water molecules forms hydrogen bond with the cellulose, okay, cellulose, cell wall, and lignin of xylem vessel. Okay. Okay. Because of cohesion and adhesion, it allows for a continuous movement of water up the xylem. It's not going to be disrupted. And of course, that tension, okay, the idea that tension is because of the, okay, upward pulling force. Okay, okay, due to transpiration at the leaves. Okay, so that's quick revision on cohesion tension theory. Okay, now that we're talking about sucrose loading, okay, I think it's a very common revision that the students always tend to would like to request to go through sucrose loading. Okay, loading sucrose. But first, you can make sure, this is just a diagram that I extract from your textbook, just a reminder to ourselves on the features of the phloem. So we have on the left, this is our sieve tube. Okay, this is the phloem tube. Okay, one cell is called the sieve element. Okay, remember that one, sieve element. All right, and then on the right, okay, always that the phloem sieve tube is going to be accompanied by companion cell. Notice that it will have all, okay, the organelles, okay, that is, most of them is absent from the sieve tube element sudah. Why? Because, okay, to ensure that sucrose ataupun phloem sap can be easily transported along the phloem sieve tube. Okay, it still has small numbers of organelles, but very much reduced. No nucleus. You see, there's no nucleus di sini. All right, no vacuole. All right, and okay, make sure that you guys also notice and remember the roles of plasmodesmata. Let's take note and go back and revise on what happens during sucrose loading. Okay, sucrose loading is the process whereby we want to be transporting sucrose from the source. Okay, we know what the source tadi kan, any part of the plant that has the ability to photosynthesize, producing sugar, which will then be converted into sucrose. And then we want to move sucrose, okay, in the companion cell, and from the companion cell, we're going to be moving all of them into the phloem. What happens? So if you look here, you're going to be focusing on what happens between these two structures. All right, so sucrose is loaded from the source into the companion cell by active transport. So this is an active process. That's why we can see there's a lot of mitochondria whenever there is an active process. Okay, that means, okay, you will see this is the reason why companion cell will have a lot of mitochondria. Okay, why? Because, okay, active transport occurs which requires ATP. Okay, again, just to reassure you guys, notice companion cell, see a lot of mitochondria. Okay, right. So active transport of sucrose, what happens between the structure? Okay, we can be focusing on this. Right, so here we have the cell surface membrane of the companion cell, and this is the cell wall of companion cell. Okay, of companion cell. Right. So look at the point here. Active proton pump. Proton again is hydrogen ions, right? Okay. Driven by ATP, which pumps hydrogen ions out of the cell into the cell wall. Okay, that's apoplast pathway, which creates a high concentration of hydrogen ions in the wall. So this is the cell wall of companion cell. This is the cytoplasm of companion cell. That means what happens is that hydrogen ions are pumped out actively, okay, right, by active transport, meaning against its concentration gradient. Pasal there's already a lot of them. There's high concentration of the hydrogen ions in the cell wall of the companion cell. So there's already a lot of them, that you're further increasing more and more concentration of hydrogen ions in the cell wall of companion cell. So by building that concentration gradient, the next thing that can happen is that the proton, the hydrogen ion, and sucrose co-transport the protein. I'm going to highlight that one. Co-transport the protein. What does it do? It is an example of carrier protein which undergoes a conformational change whereby the high concentration of hydrogen ions will then move down its concentration gradient via facilitated diffusion. This is actually an example of facilitated diffusion. Tapi, what's the difference is that it doesn't come in on its own, because using this co-transporter protein, which is this carrier protein, as it diffuses down its concentration gradient from high concentration to low concentration, it also carries along with it sucrose, like what you can see here. So now more and more sucrose are able to enter the companion cell. Okay. So what happens next is that sucrose will simply be able to diffuse from the companion cell into the phloem down its concentration gradient, but take note, through the plasmodesmata. So that is through the walls in the cell walls that you can see here. Okay, so that's just simple diffusion down its concentration gradient, filling in the sieve tube element, so sieve element. Okay, now that we have a lot of sucrose here, that will then allow the next process of sucrose transport via mass flow. Okay, mass flow. So that's another story like it. Okay, so hopefully you guys are able to understand sucrose loading. And then of course the next bit is mass flow. Okay, so what happens during mass flow? Okay, to go through with what happens during mass flow of sucrose, okay, which is basically what is it that is being transported in the phloem tube. It's a mixture of sucrose and water. I remember that one. So that what creates the phloem sap. Alright, just to remind everybody, what is transported in the phloem? Not only just sucrose, but amino acids, water together, which also might be described as the phloem sap, is what's going to be transported in sucrose. Okay, so let's go through mass flow of sucrose. What's happening during mass flow? During mass flow is generated due to a pressure difference or pressure gradient that is created. But how is the pressure gradient actually created? So the first thing that needs to happen is the active loading of sucrose. Kenapa ini mesti ada? Because what needs to occur is that there has to be a high concentration of sucrose in the sieve tube element. So there's a lot of sucrose in this sieve tube element or sieve element that I wrote earlier. Okay, so if we use this diagram, we're increasing the concentration of sucrose in this sieve tube element. Alright, so when we're increasing the concentration of sucrose here in this sieve tube element, remember sucrose is solute, right? Higher solute concentration, it causes the water potential in the sieve tube element to decrease. Okay, so if water potential decreases in the sieve tube element, what follows naturally is that water from the xylem, okay, flows into the sieve tube element by osmosis. So osmosis of water occur. Okay, so because water now moves down its water potential gradient in this sieve tube element, so there's increasing in the pressure. So there's increase in the hydrostatic pressure. Hydrostatic pressure. Dari mana? In this sieve tube element at the source. Okay. At the same time, alright, what happens at the bottom? Di sini ada sucrose unloading. Okay, sucrose will then diffuse out of the companion cell. It will enter any parts or organs of the plant that acts as a sink. Somebody, it's growing parts of the plant, whether it could be the flowers, the seeds, or storage. For example, it gets stored in the root. So when you're lowering the water potential of the sieve tube element di sini, okay, that means, okay, why? Because there's less and less sucrose, alright? So there's a water potential gradient. Okay, water will flow out, okay, following that, okay, gradient. So there's hydrostatic pressure gradient, right? So that would result in the mass flow. Okay, that's basically what I have used to describe or to show what's happening di sini. Step 2, reducing water potential in the sieve element. Water follows by osmosis, okay, and then which increases the hydrostatic pressure in that sieve tube element, causing a pressure differences which causes mass flow of water and dissolved solutes. Okay, you remember sucrose, amino acids tadi, to the sieve tube from high pressure area to a low pressure area. So there's a high pressure area from the source, low pressure area at the sink. Now, why is that there is a low pressure area ataupun why is the pressure difference is maintained? Is because at the sink, sucrose is always removed from the sieve tube element. Okay, when there is no more sieve tube element, water will simply follow by osmosis wherever there's a low water potential. So as water is also removed, the pressure inside the sieve tube element is also reduced. So therefore, you have a hydrostatic pressure difference or gradient maintained, and that's how the pressure gradient is maintained between the source and the sink. That's generating mass flow.